Hi, my name is Lejla Biberic and I'm a junior undergraduate researcher in the Pro Lab. Today I will be discussing protein structure upon transfer to the gas phase. Proteins can be studied using a variety of experimental methods, some of which include gas phase techniques. Upon transfer into the gas phase, a protein may remain in its original structure or it may change in some way. And we're curious to know where in this range is the true structure of the protein. So how can we learn more about the structure of a protein in the gas phase when experimental techniques don't work? Here we decide to use molecular dynamics simulations and specifically look at the collision cross-sections, which the collision cross-section can be thought of as a protein's shadow area. And we ask the question, how does the collision cross-section of a protein change with charge state? We use methods previously determined by our group, in which we use in vacuo MD simulations, CCS calculations, and a variety of structural analyses. The three proteins we chose were β-lactoglobulin, concanavalin A, and glutamate dehydrogenase. These three proteins represent a range of mass. Our molecular dynamics results, some of which are shown here, were used to look at the difference in structures and also used to determine the collision cross-sections. You may notice that for β-lactoglobulin, there's an orange alpha helix here and here, but there isn't one present in this 9+ charge state. These were just some of the things that we observed when looking at our molecular dynamics results. For the collision cross-section, we have shown right over here our simulated results. Our simulated results match with the experimental results obtained with mass spectrometry, in that across a protein's native charge state there is a flat trend. Because our simulated results match with experimental results, we decided to continue our investigation and look at different structural analyses. The global trends are listed here, and specifically we were looking at any patterns that may have come about with these different methods. For the interior collision cross-section, we noticed a global % decrease for all proteins in all charge states. That can be seen right over here as well. The original structure is the orange mesh, and the simulated structure is the blue protein. You'll notice how the blue protein has compacted as compared to the orange original structure. For secondary structure, there wasn't a global trend. Instead, it seemed that for each protein, something different happened. For example, for glutamate dehydrogenase, in both alpha helical content and beta sheets, there was a % decrease in change, as opposed to concanavalin A, which had a % increase. Currently, we are studying the lethal factor of anthrax toxin. The challenge is understanding this protein's conformation changes through its process, shown here, as it goes through the pore and eventually out. We are trying to understand how unfolding and charging occurs in order to better understand what is possibly going on. To do this, we're using the native charge state of LFN, a partially unfolded state, and the completely unfolded state. Ultimately, our results indicated that compaction and charge-dipole interactions appear to cancel for native protein ions spanning a wide range of mass. These findings are consistent with experimental results and give credence to structural analyses. We are currently investigating the relationship between charging and unfolding and using these methods for future predictions and interactions a protein may have with its surrounding. I would like to thank my PI, Dr. Jim Prell, along with my graduate mentor, Amber Rowland, and the entire Prell Lab. I'd also like to thank our sources of funding from the NIH, the O'Day Fellowship in Biological Sciences, along with the UO VPRI. Thank you very much.